As you heard this morning, we really seem to have reached a stage where now, for the first time actually, we have some reasonable strategies to actually begin trying to make inroads in extending human lifespan. And we really owe that to all those humble little tiny organisms, these worms and flies, that of course allowed us to do all these fantastic genetic screens that led us then to identify those conserved pathways that basically, when you start manipulating them, show you these dramatic effects on lifespan. Is that work?
Yeah.
What is it actually? Is it just slides?
Yes.
Okay, good. No, I don't think it works, Maria. This one? Ah, it does. Oh, well, don't be so negative. I think Russia also gave us Pushkin and lots of artists. Now, of course, one of the things that I think is really not stressed enough is what actually is aging. Is there, what causes this kind of processes that give us rise from this energetic young creature to this old decrepit individual? That's really, I think, still a major question because we don't know. Now, of course, we do know that people who are old have much higher chance of getting sick. There are lots of what we call age-related diseases. Now, although age-related diseases are often considered to be an essential part of the aging process, I think it's also important to actually distinguish age-related diseases from what we can call intrinsic aging on the right. Now, because this is a genetics conference, I just colored here a couple of genes that we can call disease genes and genes that we can call aging genes, different sets of genes. Now, disease genes are really genes that control the risk of getting a particular disease, like Alzheimer's disease or cardiovascular disease. And we all know that there are heritable mutations in such genes that, for example, give you Alzheimer's disease fairly early on, or give you diabetes fairly early on, or a cardiovascular disease. So, now, but in fact, this kind of diseases are far from universal. For example, mice do not get Alzheimer's disease and also don't get cardiovascular disease. On the other hand, it is postulated that there is a process which we call intrinsic aging that could actually be quite universal, not only very similar from cell type to cell type in your body, but probably also universal from animal to animal. So it could be basically the same or very similar in mice and humans, and also similar in flies and nematodes and mice and humans. Of course, the trick is now to find out if that's actually true. Is there a process that we can call intrinsic aging? And I think that question has not really been answered, although we have, of course, tantalizing evidence for it. Now, probably the first question that would come up is if there are such genes that control the rate of aging, intrinsic aging, and that control your risk for a disease, why actually are there variants that do give you an age-related disease or variants that do lead to more faster aging than normal? Why is that? Now, and I think Andre already gave some sort of a clue there, because in fact evolution, Darwinian evolution, is really not so interested in a long life. So there is no selection for genes that give you a long life. There's in fact selection for genes that allow you to reproduce effectively and perhaps even quickly in many cases. So what happens later in life is less interested, and this is actually what was for the first time basically discovered in the 1930s by the famous quantitative geneticist. But Brian Charles formulated this very succinctly here by pointing out that what really is going on is that there's a decline with age in the strength and the efficacy of natural selection, which basically means that when there's a mutation that is bad for you early on and prevents you from reproduction, then obviously that mutation will be selected against very, very easily. You will lose that mutation very quickly. But when there is a mutation that makes you perform very badly at late age, such mutation will probably stay in the genome because there's really no way to get it out. Because in nature, there are not that many old individuals, because nature is dangerous. So there are always like predators who are going to kill you, or there are accidents and infectious diseases. So in actual fact, there are very few older individuals in nature, and that's why natural selection doesn't simply has the capability of getting rid of such bad genes. And that's why they are there. At least that is the general consensus. Now, that whole evolutionary idea about aging led to the proposal that there's really, when you talk about lifespan, there's really a balance between investments that are made in somatic maintenance and investments made in growth and reproduction. But unfortunately, growth and reproduction, and Andre already alluded to that, they can have nasty byproducts at old age. In this case, it's probably, a lot of that is due to damage. chemical damage, somatic damage to your somatic cells. Now, somatic maintenance systems, on the other hand, they try to oppose that damage. And it's really the balance between the two that determines the lifespan. Eventually, somatic damage will lead to aging-related cellular degeneration disease. Although you can, as Bob showed us, increase the lifespan of a worm by 10-fold. That sounds like a lot, but still, they die. So, now, my interest is really in a particular part of that somatic damage, a component of that damage, and that's damage to DNA. And there are a couple of reasons for that. Now, first of all, that's nothing new. It's, I think, one of the oldest theories or hypotheses what causes aging. And this is actually a paper in PNAS, I think 1958 or so, by Leo Szilard. Szilard was a physicist. He's, of course, very well known. Now, why a physicist and not a biologist? Well, after Hiroshima and Nagasaki, people started to realize that the radiation was pretty bad for you. And they started to do experiments with enormous numbers of mice and rats, which they exposed to different types of radiation. And one set of experiments was actually published in 1947 by Henshaw. He basically, as part of his work, he also exposed mice and rats to very low daily doses of radiation. And then he reported on that, and he basically said, well, nothing happens. There's no effect. Well, there is an effect in the sense that they live a little shorter. But for the rest, their normal pathology, aging-related pathology, is really the same. So what they actually said that there was premature aging. And then when people realized that the radiation causes mutations, that this is why Szilard had the idea that actually normal aging is based on cosmic radiation that's also causing mutations, and they gradually accumulate in the genomes of your cells, and that's why you age and die. It's a reasonable hypothesis. But now, of course, we know so much more about what happens, how mutations are generated. And that's actually as follows. So you can see that here. So we can consider DNA damage as a driver of aging. At least I do that here. So you have a piece of DNA damage. It can be a break in DNA, a single-strand break or double-strand break. It can be like a bulky adduct, benzoylpyrene adduct. But that piece of damage is quickly recognized by a whole host of enzymes that very quickly and frequently search through the DNA of the genome to see if there is chemical damage. Why do they do that? Well, because damage is very frequent. There are probably as many as 10,000 of these lesions in any of your cells on each regular day. So it means you need very good DNA damage response systems, otherwise you are in trouble. Now, damage response systems can basically subdivide it in two groups. One basically comes into play when the damage is very serious, when there's a lot of damage. Now, that usually works through ATM and P53, and it can give rise to cell cycle arrest to give time to repair the damage, or it can lead to elimination of the cell by apoptosis. When the damage is so high, there's no way to repair it. Or it can give rise to cellular senescence, as Judy Campisi will point out. Now, this arm of the DNA damage response can, of course, give rise to loss of redundancy through cell depletion. And that, of course, could be a cause of aging. Now, it's fairly clear, I think, that we don't really age and die because we run out of cells. But definitely something like cellular senescence, as Judy will point out, can definitely contribute to aging. And I'm not going to discuss it, because she will. But I will mostly focus on the other half of this. And that's basically that the damage is being repaired. So enzymes are coming in, cut out the damage, replace it for a new piece of DNA. The problem here is that repair is not perfect. There are actually errors. Also, some damage may remain, may be persistent, and that random alterations in your genome can also give you problems and eventually lead to, for example, alterations in your gene regulatory systems. And also that can lead to functional decline, increased risk of disease and aging. Now, just to summarize it, because a lot of people is not so clear what types of genetic alterations there actually are. It begins, of course, with genetic damage, and that's really chemical damage. So your DNA molecule is chemically altered, and it absolutely needs repair, and that lesion, that break or that adduct can be recognized by an enzyme, so the repair is possible, and that happens. Now, of course, it's possible that the steady state level of damage, there's always a steady state level, that during aging begins to increase. For example, because your repair systems decline in efficiency. I'm not going to discuss that possibility. There's actually a talk tomorrow in my session by Alex Maslov in my institute, who will discuss this in some detail. Now, I'm going to talk more about irreversible changes in DNA that cannot be repaired, and that's mutations and epimutation. Epimutation, I mostly will discuss tomorrow also, but mutation, a change in the DNA code, in the information content of the DNA, is necessarily irreversible. Because when you change base pairs for another base pair, or you rearrange a piece of your genome to another place, how are you able to recognize that? It's not possible. So that's irreversible. It's very important because protein damage is not irreversible as long as you still have the gene to make new protein. But when you really have a mutation, a change in the DNA code is irreversible. And since we know that DNA repair systems are not perfect, and why should they? Because as I just pointed out, evolutionary logic basically tells you already that you will have the repair systems, the prevention of mutations, until the reproductive age, and what happens afterwards is less important. So we already know it's not perfect. That's why it's a fact that mutations accumulate during aging. I'll show you in a minute that actually it does. So the only way to recognize a mutation and get rid of it is by selection. When there's a mutation in your germline, and of course 95 or 99% of all organisms on Earth are unicellular, so basically your germline is the same as their somatic genome, they will basically get rid of that by simply selecting, so you get rid of those individuals. Now, in our body of metazoa, there are perhaps certain cell types that also can, of course, where you can have selection, but most of the time in postmitotic cells, for example, that's impossible. So that you can already imagine then that accumulation of mutations is then not unlikely at all to cause a lot of problems here. Now the same is actually true for epimutations, because you can imagine epimutations, that's for example modification of your DNA bases by for example methyl groups on your cytosine, or histone proteins that cover that are in your genome. They can be modified also, and those actually are informative, so they're really part of the informative code. But when there's chemical damage and the enzymes have to go in, cut out a piece of DNA, they also remove the layer of epigenetic control, and that also has to be replaced, and that can actually also not happen without errors. So it's the same thing. Epimutations and mutations are basically the same thing. Probably epimutation rate is far higher. I'll also talk about that tomorrow. So, okay, just to give you a little bit of a flavor of how bad this problem really is. And I sort of summarized here. This is based on a paper by Alec Morley in 1996, where he used HPRT assays, selectable marker, and you can use it for human lymphocytes, also HLA. It's a little bit of a clumsy assay, but at least it gives you pretty good ideas of what the mutation frequency is, a spontaneous mutation frequency. And he calculated this is about 1 per million per year per locus. That's how we calculate that. It seems like very, very low, of course. It is. But on the other hand, assume now that the locus, a typical locus is 1 kb, so 1,000 base pairs. Then when you're a centenarian, then each of your cells would have accumulated 10,000 mutations. So that still perhaps sounds very little to you, because our genome is really big. It corresponds to about 600 mutations per genome. But now, if you assume that we have 10 to the 13th cell, we probably have closer to 10 to the 14th, but I'm conservative here, that you can see that when you're a successfully aged human individual, then you would have accumulated the actually stupendous amount of this actual number of total mutations spread all over the genomes and all your cells. That's a huge amount. The question, of course, is what does it mean? We don't know really. Now, one question that you probably already asked is, okay, if that's true, then why didn't natural selection actually take care of that? Why didn't the mutation rate evolve to 0? At least in the germline, we already learned that in all tissues it doesn't matter so much, because natural selection doesn't work that well in all tissues. But at least in the germline, why can't it do that? Well, it's obvious why not. Because again, here you will see that mutations, although we think they're bad, and generally they are, they're actually very good for us. We would never be here without mutations. Because mutations actually, they allow organisms to quickly adapt and respond to altered environmental conditions. So without mutations, all our alleles would be fixed, and there would be no life. So life and mutations is hand in hand, it's the same. Now, probably, this may actually be an important reason, but probably more important reason is that the systems the genome maintenance systems that are there to keep mutation rates within limits, to control mutation rates, are so large, there are so many genes that do that it's highly unlikely that when you start to affect that mutation rate, that you will not also affect some other aspect of fitness. And that's probably why there's not selection to do that. But again, we don't really know very much about mutations. So still a rare event, difficult to get information, which is of course why I'm going to talk about it now. Now the model that we can then draw up, and of course very hypothetical, suppose there is this genomic or epigenomic drift, so cells accumulate mutations and epimutations. How would that work? Well, this is a healthy young individual, a liver or a kidney, and the colors indicate an increased level of degeneration because of the total mutation load. And you can see that even in a young individual, there are of course already cells that started to degenerate because they have accumulated mutations. During embryonic development, mutations accumulate already. But during aging, that problem would become worse. And you would start to see that on average, cells have degenerated further. They have accumulated more mutations. Some cells are dying, others already died. And then occasionally there's one cell, a lucky cell or an unlucky cell, depends how you look at it, that has accumulated a combination of mutations and epimutations that's really allowing that cell to expand into a cancer. So in fact, this model would explain aging in terms of degeneration, but also about cancer. So it's, I think, interesting. So what's the evidence that actually mutations or instability of the genome is really a serious problem? Well, we begin to sort of see that now. This is a paper already for a number of years ago in Journal of Neuroscience where they actually looked in, this is human, and they, shortly after we got a tool that we call interface FISH, where we could look at non-dividing cells and to see if chromosomes were present or absent. They actually did that using a probe for specific chromosomes. And they used a probe for chromosome 21. Don't immediately think of Down syndrome because that is really not so important here. It was just they had the probe available as a short chromosome. And they found out, and this is like the pictures you probably don't really need here, they just found out that 4% of all brain cells in a human, 4% of all brain cells, was actually unemployed for chromosome 21. Now, if you would assume that other chromosomes behave the same, then there's no brain cell in your brains that is not unemployed, and those were fairly young individuals. So you can really see that this is not a little thing, and that's unemployed, so a whole chromosome that changed. Now, we did a little bit of work on that, and I'm going to show you only a very tiny part. This is Francesca Fagioli, who really decided to see if this was actually age-related, because that's not known. And she used not humans, but mice, for obvious reasons, a lot easier to work with. And she actually had trouble in quantifying it. She found out immediately that it's not so easy to quantify it. When you really want to look at it in a highly quantitative way, you need a system to do that, and she used FISH probes, 2 on each chromosome. And then she only concluded that it was an unemployed event when both markers did the same thing. So here you see, of course, a 2N. Here you see they lost one, and both colors are lost here. And here's a trisomy, and you see this for both colors all the time. So only then we conclude. So perhaps it's a little bit conservative, but that's basically what it is. So if you look at the number of chromosomes, you looked at many more, but I just only show a few here. Then you can see that there is an increase with age, comparison between young and old mice in the brain, the cortex in this case. Then you see that chromosome 18 really undergoes quite a dramatic increase in unemployed. But other chromosomes do not do that, although also in the Y chromosome we see it, and also actually in chromosome 7. But in another organ of the same mice, in the spleen, you don't really see that. So it's also organ specific. It happens in the brain, but not in the spleen. And it happens in one chromosome, not in the other. But overall level, you should look at it also. This is a percentage. Overall level is exactly very high, exactly as was shown for humans. So the total amount of this kind of aneuploidy is already very high. And again, for certain chromosomes, it also increases quite dramatically with age. So this tells you already that this is really a serious problem. And are there consequences? Again, we will come back to that at the end. But of course, we don't really know. Okay, one more question that you might have at this stage. Of course, you would perhaps say, look, if this is true and it's going to bother our somatic cells, why apparently doesn't all this increasing genomic instability doesn't bother our germline? Why is the germline immortal? It would be a reasonable question. Well, the answer is very simple. The germline is, of course, not immortal, meaning actually that it is as susceptible as any other cell to increased genomic instability. Now, this is an interesting experiment published in Nature Methods in 2009, where this is an in vitro fertilization clinic. So they have lots of these embryos. They have about a 25% take baby home rate or so, which means actually that 25% of their in vitro fertilization works. So they then decided to have a lot of these very early embryos. And they looked at blastomeres, individual cells, blastomeres. And here you see them. This is one karyotype for one embryo. So the numbers are the chromosome numbers. And then you see each line, each vertical line is one single cell, one blastomere. And the colors, only when it's black, it's okay. The colors indicate like deletions and so on, all sorts of copy number variation, whatever. Now you can immediately see that there is an enormous amount of this kind of instability. And this is a number of blastomeres from one particular embryo. Only 9% of all the embryos they looked at, 9% was clean. So it tells you that it is strange because they have a 25% success rate of getting a good embryo out of it. So that tells you already that even good embryos actually have a hell of a lot of instability early on. Now, why then are they healthy embryos? Well, undoubtedly because there's an enormous selection. And as long as you are still early, you have enormous cell division, large numbers of cells, you can have that selection. And that's the only thing that happens. That keeps your germline immortal. Okay, how do we look to other mutations? Well, I will just show you a few examples very quickly. The only real way until now was to use reporter genes. And I already told you about HPRT, which is an endogenous reporter. There are a lot of problems with that, and it's a little bit of a difficult essay. So we decided to make transgenic mice and transgenic flies harboring a plasmid with a LAC-Z reporter gene integrated on one or more of the chromosomes. So we can actually excise that, turn it into E. coli, and we modify the E. coli in a way that only when there's a mutant LAC-Z that cannot give rise to an active beta-galactosidase enzyme, that they survive on this selective plate. So we know the total number of plasmids that we recover from a particular DNA sample, and we count that, and then we count the numbers on the selective plate, and this related to that is basically the mutation frequency as it was in the mouse or in the fly. And we can detect mutations that inactivate the LAC-Z gene. It could be a point mutation or it could be a deletion. And it can be a rearrangement also. When there's a break here and there's another break far out here, we can still pick that up. We can detect that. But what we cannot detect is copy number variation. Just remember it. What I'm going to tell you is underestimate. Oops. So now, this will give you a sort of a flavor of how that looks like. This is the mouse model, not the fly. So it's the age of the mouse. And here you see the mutation frequency, so the number of mutant plasmid copies, like Z copies, over the total number that we rescued from a particular sample. So it's like 5, 10 per 100,000. And these two organs, small intestine and the heart. And you can see that, yes, as I predicted, mutations do accumulate with age. And they do that in the small intestine, but they also do that in the heart. And you can also see that each dot is the average of multiple assays on one animal, one tissue for one animal. So you can see that there's variation among the different animals already at young age, but there's way more variation among the older age, which is something you would expect because variation of about anything you can measure is increasing with age. And also mutations are, of course, a stochastic process, so you would expect that to happen. I have a little bit of trouble with this, but hopefully I can work myself through it. This is sort of summarizing the situation for a whole bunch of organs and tissues, from small intestine, as I just showed you, and the heart in red here, liver, spleen, testes, and brain. And in fact, you see immediately there's big variation from organ to organ. Like our most important organs, brain and testes, they really tend to accumulate very few mutations. Although when we go into the brain and we pick out particular areas, like the hippocampus, we did actually see an accumulation of mutations with age. So you actually also have to look within an organ. But definitely there's variation. And also when you look at the types of mutations that accumulate, it's also very different from organ to organ. We published that, so I'm not going to discuss it extensively. Now, this is unpublished data. Also Alex Maslov did that. And basically what he showed here is he took cells from these mice. So these are LAC-Z containing cells. So he can actually do the same trick to measure mutation frequencies and spectra in these cells. And what he did is he treated the cells with bleomycin. And when you do that, you get an enormous increase in the mutation frequency. There's no doubt about it. And he knocked down a couple of these genes that have been associated with longevity, like FOXO, and Andre already discussed it. It's like the equivalent of DAF16 in the nematode, and that's really the executor of this whole IGF pathway. So it's basically a transcription factor that's going to activate all these stress response systems. And apparently it's doing a good job, because what you see clearly is when we knock it down, then there are many more of these mutations, both for FOXO3A and FOXO4, there are many more of bleomycin-induced mutations than when it's intact. And the same was also true for another longevity gene, the sirtuin. Sirtu1 we looked at, and Sirtu6. It's really only Sirtu6 that apparently plays a role in keeping your mutations low, because we see an effect on Sirtu6, not on Sirtu1. Vera Gorbinova will discuss it with you, and I'm sure she will agree with me that this is probably true. Now here is a little bit of our fly results. Fly is a completely different organism. Easy to work with, it has a shorter lifespan, it's much easier to work with, you don't need IACUC approval and all these kind of things. This is Ana Maria Garcia, who did these experiments. First thing she did actually, because the fly we had it much later than the mouse. Now if she compared flies, whole flies, this is DNA from whole flies, with DNA from liver and heart of the mouse, then you see immediately the difference. Fly has a much higher somatic mutation frequency. So that was interesting because we never, of course we couldn't know this because nobody had ever really compared different organisms in terms of somatic mutation frequency. That was new when we first saw that. And also the types of mutations are different. Point mutations, so base pair substitutions and so on, and genome rearrangement where you can have very large translocations or large deletions or so. So really different types of mutations. You can assume that the rearrangements are much more important. They are much more toxic. Their effect is probably much bigger because they can simultaneously shuffle around a whole bunch of genes. Now flies have a much higher frequency of these genome rearrangements. It's really interesting because, of course, we don't really know why that is. Flies, of course, they live much shorter, but still, there are cells, and all cells are based on the same biological principles. So why could the genome of a fly tolerate so many more mutations and such toxic mutations better than a mouse? We don't really know that. But in fact, when we looked at flies during the aging process, we saw exactly the same as in the mouse. Only in the fly, we had the advantage that we could look at different aging rates. The assumption is that when you keep flies at lower temperatures, you can see here, then their survival curves are extended. So they live much longer at low temperature. And the other way around, at higher temperature, they actually drop dead pretty quickly. So the aging rate is affected by the temperature. That's not really something that I came up with, but that's, I think, generally assumed by now. So that gave us the opportunity to look at these flies at different age levels, chronological age, at different conditions, different aging rates. In fact, we have a measure now for the biological aging rate. And if you plot that here, in this case, the amount of life that they actually completed, then what you see is there's a beautiful correlation, and you see that clearly that mutations accumulate as a function of biological age. So no longer as in the mouse you could only look at chronological age, but here they also accumulate as a function of biological age. And you see that, although I realize it's politically incorrect, they accumulate a little faster in female flies. In our genetic background, female flies actually live shorter. Okay, we can manipulate it. We did it in the mouse also, but we published it, so I'm not going to discuss it. But in the fly, I thought this was interesting. It's also more recently published. We actually, together with Mitch McVeigh of Tufts University, he had made flies that had a knocked out bloom. Bloom is the Bloom syndrome, the premature aging syndrome. So, and also bloom is involved in a process to optimize recombinational repair, the repair of double-strand breaks, and to sort of prevent rearrangements to occur, errors to be made in the process. So what you see, in fact, that as you compare mutation frequencies between wild type and bloom, then clearly in bloom they are induced. So when you're muted for bloom, you have more mutations. Now, ligase 4, which is a process involved in the other pathway to repair double-strand breaks, non-homologous end joining, it really has to do the end joining at the end. When you knock that out, there's no effect. In actual fact, if you looked at the types of mutations here, then you see, as you would expect, that in the bloom mutants, all these mutations are genome rearrangements. You would expect that because that's what bloom is doing. It has to prevent that from happening. So when you knock it out, it's going to happen. You get lots of rearrangement. But in actual fact, in the ligase 4, it almost seems like there are less rearrangement. You can see that especially in the females, and there are more point mutations. Now, but the lifespan, in fact, is more affected by the bloom knockout. If you knock out bloom, you can see that immediately lifespan is seriously affected. But with ligase, especially here in the females, that's much less the case. So in fact, it's much more important if you lose your bloom, which is supposed to prevent the rearrangement, than when you lose ligase 4, which knocks out a whole pathway of repair. Now, what I thought was the most interesting thing for me, because I always learned that flies are postmitotic, they cannot develop cancer, they're a lousy cancer model. But in fact, that it turns out not to be true, because at late ages, they actually do develop cancer. Now here you see 35, 50 and 60 days that in fact in the bloom you see a fairly large number of reproductive and colon cancers in these flies, but a lot to my surprise, we actually saw the same in older flies. Of course, to a lesser extent than in the bloom knockouts, because in the bloom knockouts you have all these rearrangements and they give you cancer, obviously. And in fact, you can see that here in the females, that at old age, really, it doesn't even matter if you have a bloom knockout or a normal wild-type fly. They really develop a lot of cancers. I thought that was really interesting. Okay, now you can summarize that a little bit. Remember, the only way we looked at these mutations was on a per locus basis. Now, we know the size of the locus, so we can extrapolate to the genome overall, because we know the size of the genome overall, assuming now that everything is behaving the same as our reporter. Now, if you do that, then you can actually say how many mutations, how many alterations in a genome, in a single cell, are there. And you can see that in old, only look at the old here, it's about 200 for a mouse liver, and in the heart it's about the same. In flies it's much less. So you may ask, okay, but you just told us that flies have a much higher mutation frequency. Yes, but they also have a much smaller genome, meaning that the total number of mutations per genome is smaller. But the density of mutations in the fly genome is much higher than in the mouse. Now that means actually that a fly genome can continue to provide function in the presence of a hell of a lot of mutations. How is that possible? Well, the only explanation for that is that the way a fly genome is organized and provides function is really different from a mammal. In a mammal there are many cross-links, interactions from gene to gene. There are regulatory regions that serve multiple genes. There are many of these long-distance connections. In a fly that is known, by the way, that's not the case. So because of that increased simplicity of the fly genome, they can tolerate many more rearrangements. Okay, now, of course, I told you that we look at one reporter and assume that the rest of the genome is the same as the reporter. When I made these reporter models, I of course argued that there was no doubt that it was a very good marker and it was the same for the genome overall. But now that we have a little bit better tools, I start to doubt also, and I think we probably should sequence the whole genome. So we really want to know all possible spontaneous mutations and sequence the whole genome. And we can actually do that now, because this is what it costed to do the Human Genome Project using the loyal ABI machines, and it costed $3.6 billion. But now, with the system that we have downstairs, or in my building, we can actually sequence the human genome for 5,000. And I made this slide 6 months ago. So now with the system that we just ordered, and hopefully we'll get there in a couple of months, we should be able to do that for $1,000 for a whole genome. But of course, can we actually do that? Because the cost of sequencing turns out to be only one of the problems in measuring mutations, somatic mutations, low frequency events, by high throughput sequencing. Now, you know that, perhaps most of you know how next generation sequencing works, but I will show it very quickly. Now, in contrast to Sanger sequencing, you do not sequence a genome only once. You sequence a genome as short fragments, let's say 100 base pair fragments, and you sequence them at random, a very large number of short fragments. And then you basically, what you do is you get them here are all these fragments, and you start to use a tool. So we have the reference genome, because now, of course, we don't know what they are. You just sequence them, you don't know what they are. So then you're going to map them to a reference genome. So the genome of a human or a mouse or a fly, that is a consensus genome. And you do that using a tool that we call a genomic alignment algorithm. So you align all these short reads to the genome. So now you know exactly where they are. And if you do that, then you get a certain sequencing depth. So for every locus, you have sequenced a whole bunch of short reads, and they're all here. And then you can play with it on your open your genome browser and make comparisons from species to species and so on. Okay, so why can't you really use that for measuring somatic mutations? Because it's important to know, isn't it? Now, okay, suppose this is your tissue, this is your liver, many cells. Here's a mutation in this cell. Other cells have mutations also, but they're not exactly at that position. So now you mix the whole thing up, you have your DNA sample, you're going to sequence, and you get all these reads for a particular locus, and what you see, here's your mutation. This is this mutation. But you know that because I tell you that. But your tools, your computer algorithms do not know that. They see for a particular locus, there's one read with a mutation. They think that's an error, and they're going to get rid of it. So that's why you cannot measure somatic mutations. Now, the way to do that is actually by picking single cells. Because if you now have your organ, liver or a heart, you pick individual cells, you take a cell, you amplify the whole genome. And now you amplify each and every mutation that was accumulated in that particular cell. Now here, you see what you get, what I just showed you, where you have the mixture. You see what looks like a mutation here and here, but they're not. They are sequencing errors, and the sequencing error rate of an Illumina sequencer is about 1 in 100. Now, you see the same errors here. Of course, but now we see that for here in this locus, we see that about 50% of all your reads is mutated at this position. And that is when you have a random mutation, because of course only one of the two alleles undergoes the mutation. It's very unlikely that there are two mutations at the same locus in both alleles. So that's exactly what you see here. So now we know that this is a real mutation. So when you pick single cells, you can do that. The added advantage now, of course, is also that you can now really look at a variation within an organ. You can take a whole bunch of single cells and go to look at it. That is when the cost of sequencing is going down a little bit more, which you all expect it will. This is basically a procedure for us that we actually use. I should not have shown you this slide. What it basically comes down to is we did an experiment where we took cells. Drosophila cells, because they have a small genome, is relatively cheap. So we cultured those cells, exposed cells with ethyl nitrosourea, a powerful port mutagen, and then we took exposed and unexposed cells, picked individual cells from that population, as you can see here. From the individual cells, we did whole genome amplification, but we also took the whole population, extracted DNA, did not do amplification, but she started to sequence it. And then you can see in these circles, you can, when you get your sequencing results, you can put your mutations on the inside of the ring. The ring basically shows you the chromosomes, the different colored chromosomes of the fly cells. And we can actually look at point mutations and also rearrangements. And again, I don't have time enough, I think, because I usually go over time, although we are already half hour over time, so what does it matter if I go another half hour? Is it? Anyway, if you do that for ENU, and you take a typical untreated cell and an ethyl nitrosourea treated cell, then again the red dots are the mutations. This is what you see in an untreated normal cell under culture conditions. But here, when you pick the cell from the population that has been treated by ENU, you see an enormous number of red dots, and they're all ENU-induced mutations. And of course, what you cannot immediately see here is that you can immediately see where the mutations are in the genome. You can say what the mutation is, where it is, in which regions, so you can see how nice that would be when you would be able to then demonstrate aging. And here's basically, again, the example of what I said. This fly cell line is not diploid, it's tetraploid, so you expect 25% of all the reads to have a mutation. You can see that here. Now, you see, when you see a mutation here, a mutation is defined by the difference between our sample and the reference genome. This could be a polymorphism as well, of course. Our fly is not the same as the fly that was originally sequenced. That's why we have the unamplified control from the whole population. And you see this G is there also. So we know that this is just a polymorphism and not a somatic mutation. So we can do these things. And the result, as you can see that here, typical 3 control cells for the fly and 3 different treated cells, and you see what the mutation frequency is. It's a greatly elevated mutation frequency, as you would expect. And then we actually did this for the MEF, mouse embryonic fibroblast, as well, only for 2 cells, and we see a very similar thing there. So that works clearly also for a mouse cell. Now, I was really hoping that I would have our first aging results with this assay, but unfortunately it was not to be. But I will nevertheless show you very quickly what system we decided to use for that. First of all, the fly, because again, for the cost, it's just much cheaper for us to do that. Now, we decided to pick the flight muscle because, as Vladimir explained to you, free radicals are supposed to be a major cause of damage in aging, so also DNA damage. So, and this is especially high in the flight muscle. So we decided to do that, and we picked single nuclei. So we take a piece of flight muscle, isolate single nuclei, and then we use a system driven by a GAL4 system to look at a particular protein in the membrane of the nuclei. So we can identify a particular cell, because of course we have to make sure that when we compare old with young, that we have the exact same cell type. Otherwise, we look at different cells, and it doesn't mean so much. So we actually managed to do that, and we flow sorted individual nuclei in different little wells of a chip. Then we did whole genome amplification, and we did the paired end sequencing, and that's actually where we are. So I don't have the results yet, but they should come in very quickly. So we should be able now to look at a bunch of nuclei, whole genome sequences, and identify each and every mutation that's in there. So it's really very exciting, because as far as I know, nobody has ever done a thing like that. Now, final couple slides. I didn't look at time, Bob. Is there still time for that? Okay, good. Now, a little while ago, because of course you would wonder, how would we know if mutations, even if they accumulate and they're present like 2-fold, 3-fold, 10-fold, how would we know if they really can have a functional effect? Can they really cause a decline in function? I mean, it's easy to see how they can increase the rate of cancer, which of course is an age-related disease. But we cannot really understand how high this mutation frequency should be to really decline function of a cell. Now, one way that that could happen, perhaps even the only way, is simply that increased mutations or epimutations begin to affect the transcriptome. They start to randomly affect transcript levels of genes. And that eventually could cause so much trouble to the cell that it starts to decline in function. Now, to test that, you basically need to pick individual cells from old tissue and young tissue and then simply see if particular expression levels of particular genes start to fluctuate more in old animals. And that's exactly what we did for cardiomyocyte. And we published this already quite a while ago. And just to remind you, when you have a young mouse, these are individual cardiomyocytes, each dot is the expression level, and an old mouse, individual cardiomyocyte, and you see that the difference from cell to cell, the cell-to-cell variation, is dramatically increasing, or actually it's increasing from between young and old. And that is true for, in fact, any gene we picked. We didn't pick that many genes, but like 9 or so, but those are partly heart-specific genes that we picked, and really housekeeping genes. And we see this, in fact, for almost all genes. So there is this increased fluctuation. Again, it's still not a proof that that's caused by accumulating mutations, of course, but still, it's something. Now, we became interested to see if this would apply to senescent fibroblasts. Because, as you will hear later from Judy, cellular senescence is, apart from the fact that it's an important defense system against cancer, it's most likely also a contributor to degenerative aspects of aging. And you can see here what a senescent fibroblast look like, a much larger cell, as compared to this vigorous, young, actively proliferating counterpart. Now, we actually, in this particular paper, we also looked at mouse embryonic fibroblasts treated with hydrogen peroxide. And you can see here that after treatment with hydrogen peroxide, these are the untreated MEFs, these are the treated, we see the same phenomenon. We see this increased fluctuation. This is why, of course, we said it's tempting to conclude, or to suggest at least, that what happens is this hydrogen peroxide induces random mutations, and they are responsible for this increased fluctuation in expression level. But in this case, although I realized that hydrogen peroxide, of course, induces cellular senescence, we did not immediately make that link. So Lola McRae, student in the lab, she decided to actually look at that using methodology developed by Rob Singer at Einstein. He has fantastic imaging systems, and he developed an RNA FISH procedure where you can really visualize in the cells here individual transcripts, individual messenger RNA molecules. So you see here, this is a particular, not just a normal gene, this is actually a fusion gene between an MS2 sequence, which gives you lots of hairpins. So it's an actin gene merged, fused with this particular hairpin structure. So you get many, many, many targets to bind your FISH probe. So the signal-to-noise ratio becomes better. So initially he used that for it. So we thought, well, he has the mouse, so let's take a look. So we basically did that, and you can see that what he said really worked beautifully. This is the nucleus, and you see these 4 transcription foci, because this is how genes work. You see this burst of transcription, and then it leaves the nucleus and it gets into the cytoplasm, and you see all these individual molecules, and this is basically how this FISH system works. Now, when Lola started to do this is an SMG gene. It's involved in RNA processing. It doesn't really matter. It's like a housekeeping gene. And she actually did untreated, this is human cells, by the way. She used IMR90 that Judy gave her, and she basically treated them with hydrogen peroxide, senescent IMR90. And basically what she sees is that there's this enormous increase also in fluctuation. And this time, of course, we see it directly using the imaging system. There are tools to quantify that. So this seems to be the case for this gene. Now, just because I asked her, just check. I just wanted to see if this system actually worked well in showing the expression changes that we know are associated with fibroblasts. And as Judy will tell you later, these are really important genes that are changed in fibroblasts. Increased P16, P21, and IL-6. And that's exactly what we see. In the senescent cells, using this FISH assay, we do see that those genes are greatly increased in expression levels. So that sort of told me that this system actually works very nicely. And in the meantime, she looked at more. She also looked at mouse fibroblasts again, using either natural senescence or hydrogen peroxide-induced senescence. And she basically sees the same thing. This is the correlation, the coefficient of variation. And you can see that also in this case, for this particular gene, she sees the same thing, increased variation. She also sees a decline in overall expression. Of course, you can also have the average levels of gene expression. And she also did this for the same genes that we used to work with for the cardiomyocytes, beta 2M. And basically, as you can see, this coefficient of variation is increasing for these two genes. So she basically confirmed that this phenomenon is indeed associated with cellular senescence. And then, of course, since cellular senescence is associated with aging, one may be linked to the other. Okay, now finally the conclusions. Now, first of all, I think it's pretty clear now that the genomes of somatic cells are really very dynamic. It's definitely not true that they are static and cannot accumulate mutations. They accumulate lots of changes, as we demonstrated, all types of changes during the lifespan. Now, we also show that the high throughput sequencing can actually effectively be used to comprehensively characterize the genome for this kind of events, but only when you pick the single cells. This again has the added advantage that you can now also look within an organ or within a tumor to look at intra-organ or intra-tumor variation, cell-to-cell variation, which is not unimportant. And finally, it might be the case that actually random genetic alterations may cause aging through an increase in what we call transcriptional noise. Now, the people who did the work, I think already gave them, Francesca Fagioli, the aneuploidy work. Sylvia is coming tomorrow with some of the epigenomic work. I did not show a picture of Wenji Lee and Michael Gundry, unfortunately, who did the work with single-cell whole genome sequencing. I don't know why that was lost. Lola, I did show that to you. Alex, I mentioned it a couple of times. And Brent is very important for us for the data analysis. And here are my collaborators. Thank you so much.
As I have understood, you performed a single-cell sequencing of mouse tissues and flies for mutations.
Yes.
So about 50 mutations in the whole genome of mouse liver and 3 times more in old age. So my question is, how did you investigate different cells, and how frequently do those mutations repeat? Oh, this is completely random mutations.
Well, with the whole genome sequencing, does it work actually? It doesn't work? Okay, I'll use this. So with the whole genome sequencing, we have not yet looked at old versus young. We only looked in cells, treated or untreated. But the variation from cell to cell in the total number of mutations was not very big, as you could see. It was the same for the small variation, both for the fly and the mouse cells. But of course, they were cultured under identical conditions and probably close to each other, so it's not so surprising. The mutational spectrum was different, completely different. There was a certain prevalence of certain types. It's all in the paper, you can read it. There's a certain prevalence of types of mutations, but that's not unusual. You would expect that. Mutations, you see particular types of mutations more frequent than others, which really have to do with the gamma-call that will damage the DNA. It's simply these gamma-calls have particular specificities, but overall the pattern was really almost completely random.
Well, thank you again for your brilliant presentation. I'm going to ask the question which is more or less far of your particular field, but nevertheless, I'd like to learn what happens with mitochondrial DNA synthesis. Mitochondrial DNA synthesis. We know that aging is always accompanied with a very large increase in the amount of mitochondrial DNA, like in apoptotic cells. Can you have any idea what it's for?
So your question is about the mitochondrial DNA?
It's also part of the genome.
Yes, I have to say we did not look at mitochondrial DNA because again our reporter assay is in the nuclear genome, and in the next generation sequencing, of course, we see a lot of mitochondrial fragments among the reads, But unfortunately, you have many mitochondrial genomes per cell. So we would not be able, again, to distinguish mutations in the mitochondrial genome from random errors. So I cannot really tell you anything about what happens in the mitochondrial genome. But the one who has done a lot of work on that is Konstantin Krapko, and he published really a lot of papers on mitochondrial mutations. This actually is not as straightforward as it seems. It's not so easy to measure mutations in the mitochondrial genome. But in general, of course, it's known that there's an age-related increase in the mitochondrial genome. I actually didn't know that there was a loss of mitochondrial DNA during aging. I didn't know that.
Eva Sikora, Warsaw, Poland. I'm here. How would you explain the origin of aneuploid in the brain? Stem cells or mature?
Yeah, that's an excellent question. I didn't say that. We actually later checked what cell type was associated with the aneuploidy, and we used a NOI-N antibody to Vijg sort only the neuronal nuclei. It's the same trick. You can basically isolate nuclei from the brain, and then you Vijg sort nuclei using an antibody for NOI-N, which is also in the nuclear membrane. So we could identify then that all the mutations were outside the neuronal nuclei. They were really probably in astrocytes or so. Most of the other cells, glial cells, are astrocytes. So they are not in the neuronal cells. That is to say, originally, at young age, you do see them, say the sort of 1% of the brain cells contain, are aneuploid, irrespective of the cell type. So also neurons have their share on aneuploidy very early on. But that, of course, you can easily explain by early development, so then they are undergoing cell division. But later in life, there's no aneuploidy accumulating in neurons. We really see them virtually limited to the glial cells. And that, you basically already suggested that, is obviously because those cells still can undergo cell division, and aneuploidy must be due to errors during mitosis in the spindle checkpoints, for example. So it's a very good point.
Igor Artyukhov, Institute of Biology of Aging. I have a question that consists of two parts. First, how can germline DNA avoid mutation in genes that are active only on later stages, such as related to reproductive system or any others? And how can they avoid the catastrophe of collecting mutations? And other one, it seems to me that only mutations in germline DNA are related to evolution. So they may be of some value, but not mutations in somatic DNA that can be only harmful. Isn't it so?
Yeah, I mean, the last question, of course, you can have actually, there's some evidence now that there can actually be transgenerational changes. Actually, there is a little evidence that mutations in somatic cells, in fact, can have an effect on the germline. Anyway, that's a tiny thing. Now, your first question, how can the germline survive? I think I addressed that. It's basically by selection, because it's well known that there are, for example, only probably 30% of all conceptuses really survive and you have a live birth. It's only 30%. And when you look at those spontaneous abortions, then they're flooded with chromosomal instability. So,
but how can there be a selection in genes that are not active on this stage?
How can there be a selection? You can basically, oh, you mean the actual mechanism that they, for example, that there's apoptosis or so. Is that what you're talking about?
If a gene is active only on later stages of life, then it is not active while pregnancy, and it cannot be aborted.
I don't know. I don't think I understand the question. Perhaps we can talk about it later.
I think this is a discussion, and you can discuss it
later.
Yeah,
that's what I think.
If I am not mistaken, there is some information about calculations, quantitative calculation of mutation rate in mouse and human cells. As I remember, it's some very old data, but it reveals that there is a similar frequency of genetic mutation, but epimutation is much more frequent in mouse. It means that epigenetics is more important in aging than genetics, or it's not so.
Yeah, no, that's an excellent point. I think I also briefly mentioned that it's highly likely that epimutations are much more frequent than mutations, but there's no evidence for that except from Arabidopsis. There were some recent papers where they basically looked at that in Arabidopsis, and they actually found that there was a very high, I mean, Arabidopsis is a plant, so it's actually, you can sort of look at epimutations more easily as sort of germline mutations in the meristeme. So they found that it was far higher than sequence changes, exactly as you would say. Now tomorrow I will actually talk about a single cell assay for looking at DNA methylations, and I will then show exactly what you suggest, that the epimutation rate is far higher than the mutation rate. So you're entirely correct.